We shall now start the scientific part of the meeting. So it has become more or less of a tradition to start this meeting with Professor Jean Clairambault. It's an unwritten tradition. Professor Clairambault is also a medical doctor, and he has been a tremendously active person in this area of connecting mathematics, biology, and medicine. And he's going to tell us a few words about tumor growth. But before he starts, I'm afraid I will have to make some announcements. As we can see, we are a little bit behind schedule. So for the morning talks, we are going to run 30 minutes out of schedule, meaning we are going to shift 30 minutes up. So Professor Clairambault is going to start now, and he is going to have the usual 1 hour. So let's say 50 minutes plus 10 for questions. And okay, so thank you very much, Jean, for being here, and it's really a pleasure to receive you again.
So thank you very much, George, for the nice presentation, and thanks to everybody in the invitation organization committee for inviting me, and especially to give me the honor to begin, which actually happened already once or twice. I don't remember. Really? Well, I remember a time when there was the first talk given by Fields Medal. That's not my case, unfortunately. It was Stephen Smale, I think, sometime in Angradus Race. But just the same, I think I am the oldest now, so maybe it's the reason why I am beginning. So I will talk, as indicated in the title, about tumor growth with a special focus on drug resistance and the issues this gives about therapeutics, which are always present and more present in cancer therapeutics, but not only in cancer. I would just the same focus on cancer. But it's also a matter of bacteriology, antibiotherapy, virology, etc. And all this comes now with an evolutionary view. And I'm very glad that Mark Souza presented the, let's say, the auspices of the conference under evolution, because it's more and more important and seen by biologists and physicians also as important. That is not only considering tumor growth and therapeutics from the point of view of one cell and acting on one specific intracellular pathway, but also all populations of cells and interacting cell populations. So that's in the middle, in the absolute middle of evolution. So an evolutionary view which may have also impact on therapeutics. And I will also present open questions about modeling that are more and more challenging and that we are addressing with mathematical views. So what I will present is certainly not only my work. It's a work of a team that is working at INRIA, National Institute for Research in Control Science and Informatics, and also at UPMC, Université Pierre et Marie Curie, Paris, at Laboratoire Jacques Guillons, which is headed by Benoit Pertame, mentioned already, and very important in organizing this conference up to today. So my team, my INRIA team, is called MAMBA for modeling analysis for medical and biological applications. And this is what we are all in MAMBA dedicated to. So a bit about what I will talk about to sum it up. Intratumor heterogeneity is a very important thing to have in mind when you try to address tumors and cancer cell populations in general, because it is absolutely necessary to consider this heterogeneity and to code it in mathematical models by coding variability or heterogeneity, which is the same, by different means, and in particular by structured equation models, that is PDEs, but structured not only in space, but also in phenotype, space of phenotypes, according to the importance that you give to such and such a phenotype in the particular problem you are addressing. So slow genetic mechanisms are certainly at the heart of evolution from billions of years, and this is important to consider because you may be certain that the ability of cancer cells to develop resistance is due to accumulation of mechanisms or mechanisms that are hidden but may be revived in our genome from the very ancient times of planet Earth until now. And it's not only these slow mechanisms that are at stake, but also very fast mechanisms that are responsible for the stress response of cancer cell populations that are confronting very highly aggressive drug therapeutics. So this is what I will develop in the first step. This way of cell populations being reacting to very aggressive therapeutics, cytotoxic therapeutics, may be due to the fact that they are plastic. These cancer cells are very plastic and able to revive hidden mechanisms that are normally not present, not expressed in our phenotypes in our genome. So an important thing also to consider is the fact that such plasticity in cancer cell population may be reversible. That is, when it's not due to genetic mutations, when it's only due to revival of genetic mechanisms that have been hidden by epigenetic mechanisms, then it is reversible. And I will show you an example of biological observations and also of how we may model this. And I had presented it already 2 years ago, but I think that it's still important to have it in mind. So our models also, that is structured population models, have the advantage to be amenable to control by mathematical means that represent different kinds of therapeutics that are exerted on cell populations. And finally, I will present modeling questions that are very challenging and that we may formalize in mathematical terms. And that's what we are trying to develop nowadays. So drug resistance, as I said, is a thing that is present in many medical situations, not only cancer, but parasitology, virology, antibiotic therapy, etc. Usually, due to diseases due to pathogens that are not usually present in cancer, because in cancer, it's only deregulation of control mechanisms, and in particular of control of differentiation, that are at stake. But just the same, you have the same issue of population of individualized living organisms that is confronted to an extremely aggressive environment and is able to react to it by developing special subpopulations that are able to resist and develop the population after some time. So drug resistance also is important to consider because you may remember that maybe in the 20 or 30 recent years, the emphasis had been put on targeted therapies in cancer that may have some successes, but very often go to failures. Why? Because usually targeted therapies address one or two specific intracellular pathways, but at the level of one cell, not of the cell population. And this may explain why there are so many failures. And also, a thing that may be mentioned is that maybe this stress response, in fact, has been coded in our genome for quite a long time, and this has been exemplified in this article of PNAS 2005, emphasizing these so-called cold genes that are very ancient in evolution and are able to make cancer cell populations and plastic cell population able to resist aggressive environment. So formally, the accent, the emphasis was put on molecular mechanism, but as I said, this goes to failure, because if you don't take into account the whole cell population, then you're done for. You won't be able to have access to the evolution of the population towards drug resistance. So it's not only the mechanism, the individual molecular mechanism that's at stake, it's what happens at the level of the cell population. And you may consider Darwinian selection, of course, Darwinian selection of the fittest, but as you will see, Darwinian selection goes on a slow pace, whereas drug resistance to very aggressive anti-cancer therapeutics are very fast. So it's very likely that not only Darwinian selection, but also Lamarckian adaptation must be present. So just to illustrate this idea about the ancient genome that we have in our, the ancient mechanism that we have in our genome, in fact, it has developed with more and more mechanisms up to the level, beginning from the last universal common ancestor, which was a unicellular organism, up to now, vertebrates and in particular mammalians, with many, many different mechanisms inscribed in our genome that make us, when these mechanisms are revealed, able to resist extremely aggressive therapeutics. And as Mark Vincent, who is an oncologist in London, Ontario, said, cancer is more a matter of an archaeoplasm than of a neoplasm. This has been popularized recently by physicists, Lineweaver Davis and Vincent. And now, after this physicist's view, biologists are trying to show that there is some reality in these views. So to make the long story short about the atavistic hypothesis of cancer, when you see this timeline from the beginning of life on Earth until now, until us, the main idea is that the genes that have appeared in the process of development to multicellularity, because of course cancer is a disease of multicellular organs, that is metazoa if you want, only, not of unicellular organism, then these genes are the same that are altered in cancer. So the correspondence has been always, not always, recently only addressed by biologists in particular, they're not mentioned here, but Domazet-Lochot and Trautz, who are trying to reveal with phylogenetic, phylostratigraphic studies the importance of these genes that are important in the development of multicellularity with respect to the fact that they are altered in cancer. In fact, you must think that evolution, as François Jacob in a celebrated article in 1977, I think, in Science said, evolution is related to tinkering. That is, evolution does everything it can from what is available in the environment. And sometimes this tinkered mechanism may go astray, and this is particularly the idea that we have when we say that the altered genes in cancer are those that are responsible for the development of multicellularity, which is a very complex phenomenon and mechanism, of course. In this way to evolution to multicellularity, I have this representation that is also confirmed by studies in acute myeloid leukemia, that first it begins multicellularity by very simple things like proliferation and apoptosis control. Then you have division of labor and you have differentiation in different kinds of cells. And finally, to control differentiation, the epigenetic control comes and makes things such that the organism is coherent. But the first thing that seems to appear in cancer is the failure of this epigenetic control that may take years, but then goes to failure of differentiation, of transcription factors, and then finally to defects in proliferation and apoptosis control. But what is important also is the fact that when we are addressing by anti-proliferative drugs cancer, we are addressing precisely the cancer where it is the strongest. So it's a comfort to think that nowadays immunotherapy is going strong and may address things that are not able in cancer cell populations to resist, because it's a much more advanced mechanism of control, the immune response, than just proliferation and apoptosis. Well, why resistance in cancer? And here I will briefly mention the works of Sui Huang and Stuart Kaufman, and about epigenetic landscapes that have now, there is not only a metaphoric meaning, but also some kind of meaning with respect to systems biology. As Sui Huang said, it's not only the fact that biology does not make sense except in the view of evolution, it's also the fact that evolution does not make sense except in the meaning, in the view of systems biology, which may be a bit extreme, but interesting at least as a view. Also, a thing that we have not addressed yet is the fact that tumors are not just aberrant distributions of cells, but when they are successful, they represent cooperative subpopulations of cells and the beginning of division of labor. So that's a thing that has been addressed by biologists, but to my knowledge, not by mathematicians nowadays. What I will insist on is the fact that the basis cancer cells is very plastic, that is very able to de-differentiate, to go to less differentiated states, and is capable of adaptation to very different environments, and in particular, very aggressive environment, as it has been the case in a remote past of our planet, that is acidic environment, low oxygen, high ultraviolet radiations. And all these mechanisms have been likely stored in our genomes and may be revived by cancer cells and their plasticity. Now, well, this is just to mention that Sui Huang has developed at a short time scale, not the long time scale of evolution that I presented in the previous slide about timeline, by this epigenetic mechanism. And as I said, this is a reversal of the Dobzhinsky motto by Sui Huang recently in one of his papers. And the idea is that the epigenetic landscape, starting from one genome, here you have a different view from what is adaptation of genomes in a global environment. You have adaptation of one genome towards its diverse phenotypes, that is, diverse differentiated cells, to go to different tasks normally, and if it's a cancer cell population, to go to different tasks for resistance to drugs. So in cancer cells that are very plastic, it may go very, very fast. So this is also what is very much well known now. It's about this epigenetic landscape, but for a very special case, which is hematopoiesis. In this case, the different paths are very well known and have been illustrated, and also some systems biology models have been proposed to address this block. For instance, there are blockades at some stages here which may result in acute myeloid leukemia, and this may be addressed by systems biology modeling. So this is a general attempt by a hematologist, Tariq Enver, who is in Britain, to represent, but of course in a very speculative mode, what may be from one original single cell, the original cell that we all came from, towards differentiated cells in physiology, and with at some nodes some facts that may be represented by mathematical equations, usually ODEs, but also PDEs may be used in this setting, and this has been done by Avner Friedman in particular. Now, biology. Well, can drug resistance be addressed by biological experiments? We have, since the founding experiments by Luria and Delbrück, this alternative is when a population is able to resist, is it due to the confrontation with the agent, with the pathogen or the drug that addresses and puts the population in a deadly situation, or has it been developed by different mutations according to the development of cells? And then there's selection, Darwinian selection. And in fact, the experiment of Luria and Delbrück was very simple, taking different petri dishes and putting different, these are bacteria, not cancer cells, and for sure bacteria are less smart than cancer cells. They don't have all this genome storing that I mentioned before. And then confronting these bacterial populations with phages, that are viruses that eat bacteria. Well, if it is only confrontation with the phage that make some of the bacteria resistant, then in all petri dishes, I don't know, for instance, here there are 4 represented, it may be 30, you should have the same situation. Selection of a resistant population at the same rate. Whereas if it Darwinian selection, then you may think that some selection has occurred with the time of different divisions of the bacteria, and then you should have different situation. For instance, in this case, very, let's say 50% of the bacterial population resistance, here nothing, here very few. And so this was a test, a biological test. What is the real situation? And the real situation is always the second one, that is pure Darwinian selection in bacteria. So this is challenging for our vision of resistance in cancer cells. Is it the same? And we will show that it's not the same. In particular, because it's a matter of not only selection, but also of adaptation, and as I said, reversible adaptation. And our first modeling came from this article, which was published in Cell in 2010, which shows that very aggressive cancer cell populations, non-small cell lung cancer cells, in fact, exposed to very high doses of drugs, finally resulted in resistant population that took over. But when you withdrew the drug, then this resistance came to extinction, and the initial population developed again. So it's one of the first occurrences where it's shown that this resistance is reversible. To make this sum up of the Sharma paper, they took a very aggressive population of cancer cells, exposed them to gefitinib and then other drugs at a very high doses, 50 times the doses that are used in clinic. And then what they observed is that most of these cells die, only 0.3% who were surviving. But after some time, these surviving cells that were at the beginning not proliferating began in the same aggressive environment to proliferate again. And then they called these two different subpopulations evolved in time. First, drug-tolerant persisters and drug-tolerant expanded persisters. These names come from bacteriology, but it was absolutely the same as observed in cancer cells. And I don't know about bacteriology, but what is important is that when you withdraw the drug, after some time, depending whether you are the deep TP or DTP stage, then there was resensitization, complete resensitization after some time in the cell population. That is, they were able to be attacked successfully by the drug again. And you begin the scenario, the same scenario. So, and this was a very nice work by our postdoc, Tommaso Lorenzi, who was 2 years ago in Cabo Frio with us, and Rebecca Chisholm, who are now both in different positions, one in Scotland, the other in Australia, to represent this by a PDE situation, by an adaptive dynamics modeling. So the first idea was to forget here the red and the blue term, to put a non-local Lotka-Volterra reaction term representing selection. I say non-local because here you have the fact that this proliferation term here is dependent on the total population. This is a proliferation term. It depends on two phenotypes that are supposed to represent the heterogeneity of the population. Normalized expression level of survival, if you want viability, and another one related to proliferation, if you want fecundity of the individual cell in the PDE model. So if you forget about the red and the blue term, then you have a pure non-local Volterra selection term that represent Darwinian selection. Non-local because, as I said, the proliferation term here depends in Lotka-Volterra mode on the whole population of the cell. So first attempt didn't work, did not manage to represent the observed mechanism that was presented in the biological setting. Then they had added Laplacian, that is something representing non-genetic phenotype instability. Non-genetic because we know that it's reversible, so it may not be due to gene mutation. And then they observed something going to in the sense of the observation, but much too slow. Because this, if you have just this term, then you represent Darwinian selection at a very, very slow scale. Then you have to represent adaptation to the drug environment, and then they added this stress-induced adaptation, which represent Darwinian adaptation. And this was dependent on the dose of drugs, and also there's a logical variable here, V bar, which represents the fact that we may have this model without adaptation, V bar equal not, or with adaptation, V bar positive, with no flux boundaries for the whole population model. And with this model in mind, they had in parallel the development, this was mainly what Rebecca Chisholm did, of an agent-based model. And here it's easier for us and for biologists especially to understand what is at stake. That is, first, selection of a small subpopulation, then this small subpopulation is developing, proliferating again, so DTPs and DTEPs. In a sort of phase plane diagram, you have the evolution time is just mentioned by arrows from the initial site population with a drastic development, decrease of proliferation and low survival at the beginning, but just maintain survival. And at some time in the evolution, re-increase of proliferation, then initial cells, DTPs, and finally DTEPs, with a parallel representation of the evolution of the average proliferation potential Y and the average survival potential slowly increasing. So with this agent-based model, they paralleled this representation and obtained, for instance, this was a movie performed by Rebecca, which shows you in the agent-based model what comes on. Oh, very, very few cells at the DTP stage, then proliferation again. And this was published in Cancer Research 2 years ago and was very interesting, or judged so, very interesting by the biologist who saw it. So let's, let me see. Maybe, yes. I think it's stabilized. Thank you. So the, maybe, no. Again, sorry. No, it's not biology. How did I get it? I think I missed a slide, but never mind. It's okay. I think it's okay now. Yes, this one. Thank you. I was trying to find a slide that maybe I forgot to put. Well, this is just to make sure that when you have the agent-based model and you take the average, you recover the same thing as in the PD model, but of course, agent-based models are just very different. Yes?
Just a question. You were talking about these non-genetic elements.
Yes.
You are talking about these non-genetic elements, but you are basically talking about epigenetic elements.
Yes.
Can you consider also cytoplasmic elements like plasmids and things like this? Because they are important for drug resistance, for example.
Cytoplasm mechanisms. Maybe something happens in the cytoplasm, but according to what we have seen, because what I have not mentioned also is the fact that this reversal is completely blocked when you block an epigenetic enzyme. So that's right. I had forgotten to say that. And people might say, well, it's a purely physical mechanism, nothing to do with epigenetics. But the important things that I forgot to mention is the fact that the reversal doesn't work if is blocked, I mean, when you block an epigenetic enzyme. So maybe there are other mechanisms, and it's very likely. But the epigenetic mechanism is certainly very important. And we know that epigenetic mechanisms are reversible. So they are very likely candidates to this explanation of the reversible drug resistance mechanism. So this was just to mention that it's different at the agent-based model level because you may have completely different behaviors. And the only interesting thing is the fact that the average of population gives the same result as the PD model. So I think I'll have to speed up a little, but just the same, it's important to notice that the reversibility is coded in the model. In fact, our model is not unidirectional. When you withdraw the adaptation term that's due to the drug, that is when you put the drug concentration to 0, then it's completely reversible. And from the development of this, at any time when you withdraw a drug, you reverse towards completely sensitive mechanism by withdrawing, by washing out the drugs. But we have two possibilities to explain this in principle. The pure Darwinian scenario with no adaptation, that is, as I said, V bar equals 0, or the Lamarckian scenario with adaptation with V positive. That is the Luria-Delbrück scenario A, that is the hypothesis that was proved as impossible in the case of bacteria. And the Luria-Delbrück scenario B, the one that was proved by Luria-Delbrück in the case of bacteria, that is pure Darwinian scenario. And in what we've designed in our simulations, we've always thought of addressing the two scenarios. Also, it's possible to mention the fact that heterogeneity is a thing that evolves. Here, it's evolution without drug. So you can see that starting from 0 here with a sort of a Dirac-like distribution of the survival or of the proliferation potential, you normally go to a sort of a Gaussian-like distribution. But if you add drug treatment, then it's completely different because you're starting here from a very a non-resistant cell population with a survival potential, is it? Yes, here at the middle. Then you obtain a survival potential in the end, which is very, very important in the cell population. And as regards the proliferation potential, it is a bit different because starting at the beginning here, you go to, is it here? Yes, with a cell distribution of very proliferative cells, you go to a non-proliferating cell population, and after some time, you go to a mid-state of a proliferation phenotype. So this is what may be addressed by the model. And we asked with this model 3 questions. Is the Laplacian term in the PD model, that is non-genetic instability, crucial for the emergence of DTEPs and DTEPs? And what can we expect? Second question, if the drug dose is low. And finally, can genetic mutation explain this? Well, the answer, whatever the scenario, to the first question, is the instability crucial for the emergence of DTEPs? Yes, always yes. As regards low drug doses, it results in DTEPs, but not in DTPs. That is, the transient population is not seen in the case of the low drug doses as with respect to the other cases where you have very high drug dose exposition. And could genetic mutations, well, we took a mutation kernel to explain this, did not explain this. Well, this is what we did by our simulations to show this, what I was mentioning here, but I will not go in detail just to keep time to address the rest, just to mention that this is what we did to address the case of mutations compared with pure mutation model compared with our model. And whatever the case, it was not possible to obtain the same behavior. So now, to sum up our results, if there are some DTPs present, that is in the case of the pure Darwinian selection mechanism, then you must have the non-genetic instability, that is the Laplacian present, with, of course, the selection, that is the reaction term. If no DTPs are present initially, you cannot explain this by pure Darwinian selection. You have to add the stress-induced adaptation that correspond to a Lamarckian mechanism. Not clear so far that there may be therapeutic consequences, but we try to address this with oncologists and biologists by trying at least to conceive, to have an idea of what could be an association of drugs, cytotoxic drugs and epigenetic drugs to block the epigenetic mechanism that results in DTPs and DTEPs, to combine them to block resistance. But this is still speculation. Now I will focus a bit on more analytical, that is mathematical settings, not only simulations, with a general presentation of phenotype-structured population dynamics. It's very simple. As I said, you have the fact that there is a phenotype or a trait in general in which the heterogeneity of the population is coded, and we call this trait X, maybe a phenotype. In other cases, it may be cell size, it may be age in the cell cycle, for instance, or anything. But in our case, it may be either resistance to a drug or, for instance, the XY phenotype by dimensional phenotype that I mentioned. So the interest of such modeling is that you can address both the distribution of the trait in the population and its asymptotics. And if you integrate with respect to the trait, then you can have access to general tumor growth. And of course, the interest of such models, if you include targets for control, is that you can develop optimal control. So, and this is the work that we have developed with our PhD student, Camille Pouchol, here, with the help of his other co-supervisor, Emmanuel Trella at Laboratoire Jules Williams. So the prototype model may be this one. You have here proliferation term, here death term. And the death term is dependent both on a trait, on a phenotype, which may be just in this case resistance to a drug, and the total cell population. So this is a non-local Lotka-Volterra model, very simplified one. So questions are, what is the asymptotic behavior of the total populations? That is, does it converge to a stationary distribution? And also, what do the phenotype or phenotypes, if we have a multidimensional phenotype, do in the population? So the first result in very simple settings of this convergence of this problem is that there is convergence to a stationary distribution. This has been shown with, and it's not very new, but it was shown in detail in Camille Pouchol's internship report in 2015 or 16. Then when you have convergence, you can ask what is the behavior of the phenotype distribution. And for instance, here you can say that starting from a resistance, if you think that here the phenotype is resistance close to 1, then with evolution, without drug, without any constraint in the environment, you reverse towards convergence to a sensitive phenotype. So this idea may be shown very rigorously as the fact that the population, the total cell population, is converging towards rho infinite, that is the level of the plateau of the stationary distribution, multiplied by a Dirac in the phenotype. And this is accessible if you want to see the proof in Camille Pouchol's internship report. Now we try to represent the same mechanism, but with inclusions of targets for drugs. So this is here the representation of the proliferation term, the proliferation rate that is without death, and here is the death term here, and an added death term here. So this is what we call cytostatic drug that impinges on the proliferation terms, but without killing cells. It only slows down the cell cycle velocity. And whereas the cytotoxic drug here impinges on the death term, that is actually kills cells. And in this setting, having both healthy cells and cancer cells, we try to, with a common drug, combination of two drugs, to address the problem of keeping healthy cells alive and extinct. These are healthy cells, these are cancer cells, and going to extinction for cancer cells. And this is what I call pedestrians optimization. This has been just simulated by Alex Lortz and Camille and Tommaso Lorenzi in this paper of 2013. But now we want to address the problem from the point of view of optimal control. And then, which has been what has been shown in an article here published at the beginning of this year in Journal de Maths Pure et Appliquée by Camille Pouchol and other authors were Alex Lortz, Emmanuel Trella, and me, is that there is certainly convergence and concentration to phenotypes, which may be not necessarily just one singleton, but a combination of different Diracs, but isolated phenotypes. And this has been done by using a Lyapunov functional. So the general problem is here you have the controls, but in this case, it's constant control, what has been first shown. So it's the same as the natural evolution, with, because controls are constants, it's like no control at all. And then it's possible to show, by using a Lyapunov functional, that there is convergence of both populations, and there's unicity of this convergence, of course. And furthermore, it's possible to show that as regards phenotype, they are concentrated on a set of points in the interval 0-1, which is the range of evolution toward drug-resistant phenotypes, that represent the distribution of phenotypes, of the asymptotic distribution of phenotypes. This has been shown by Lyapunov functional that has been identified and used by Camille Pouchol, and this is a general form. In fact, I asked different people about this. It seems that, according to physicists, it's related not to the usual energy that is used in entropy, in generalized entropy formulations, but to free energy. I'm not a physicist, so I don't see exactly where it comes from, but you have this logarithm and the difference of the two populations also that are involved in this Lyapunov functional. And this Lyapunov functional, which is just a sum of two Lyapunov functional, one addressing the healthy cell population, the other one the cancer cell population, may be managed in such a way that you obtain convergence and concentration. So this is exemplified on this simulation. But here it's exactly what must not be done, because here you give very high concentration, constant concentration of doses, and what you obtain is the fact that you are absolutely deleterious to the organism, because you select, by doing this, a very sharply organized cancer cell population that is absolutely resistant to the drug. And then you can see that the cancer cell population increases, whereas the healthy cell population decreases, and also the rate of the sensitive population in the cells decreases also. So, well, I skip this. And the optimal control problem is what we addressed, is precisely this one, with finite horizon, minimizing the total cancer cell population here, with the same model, of course, with constraints due to the fact that we want to preserve healthy cells in the total population, and that we want also healthy cells not to go too far below a reasonable part of a reasonable proportion of the initial healthy cell population. So with this, it's possible to show a theorem that is published in this paper this year with Camille as the first author. And also now comes question of confrontation to the clinic, because here what we have is the fact that we have finite horizon, which is absolutely non-clinical setting. Physicians don't use a finite horizon. So, of course, this is the right with finite horizon strategy. First, do not give anything in cytotoxics. Give a high dose of cytotoxics and then a middle dose. As regards cytostatics, give a very low dose and then the maximum dose at the same time. And then what you will observe is the fact that the cancer cell population will decrease very slow to a very low population, whereas the healthy cell population will be maintained over a given threshold. But what we have to address now is the fact that in clinical settings, it is due to settings that, due to local constraints, clinical constraints, it is in a so-called quasi-periodic setting that you are working. So we are trying now to, with the help, hopefully obtain help of the oncologists to develop the compatibility of our optimal control settings in finite horizon with other settings more closer to the fact that it may be periodic, almost periodic. So this is work in progress, and it's not easy to obtain collaborations of physicians, of clinicians on this. Limitations of this optimization procedure, of course, is the fact that we have one drug-resistant trait in a population that is supposed to be completely described in its heterogeneity by this trait, and of course it's much complex than that, and we will develop I will skip this. I will skip this also. I will skip, just to mention it briefly, that it's possible to have phenotype space and resistant and also Cartesian space, that is to, in the case of spheroids, to include, but this is published in this article of the Bulletin of Mathematical Biology 2 years ago also. It's possible to take into account spatial considerations if you have access to the geometry of the tumor. If you don't have access to it, representing spatial constraints does not make sense. So this is, these are my references. Do I have 5 minutes more or 2 minutes? Okay. So I'll try to represent now very fast questions that we are addressing that come from biology and from evolutionary biology, and that must be involved in future mechanisms. In particular, I will mention an important thing. I mentioned this thing about phylostratigraphic studies, evolution and tinkering, phylogenetic scenarios of evolution, etc. But I will mention mainly the fact that bet hedging is a very challenging way of the tumor cell population to face exposure to very high doses of drugs. And we may think of it as a distribution of phenotypes that is not one just resistant phenotypes of a population that advances very regularly in a phenotype space toward more and more resistance, but maybe a distribution of concentrated phenotypes in a phenotype space with development of specific resistance towards such and such a phenotype. And the phenotypes we are addressing presently are two of the phenotypes I mentioned, fecundity and viability, but we must, I think, address a phenotype that would be plasticity of cancer cell. That is the ability to develop dedifferentiation. And it seems that this bet hedging is very important in the representation of evolutionary mechanisms in other parts of evolutionary biology. And I think it is also something to be considered And we will try to develop this in, I think, models that are close to the ones I presented, but with additional things. In particular, there should be, with respect to the optimal control thing, that is the work of Camille Pouchol with Menetrella and Alex Lortz and myself, just reactions. We must include diffusion, and we must include also advection. And with this, we may obtain very concentrated and very fastly obtained concentrated phenotypes. So many other questions, and these are more for biologists. And a few articles that have open way. We've begun to, with other colleagues, to develop reading and developing models about this. So I will just, I think, yes. Recently, I have presented with other, and in particular with these ex-postdoc Rebecca Chisholm and Tommaso Lorenzi, the fact that there are things that are of biological origins that can be represented partially by a mathematical model. But I will not go further on this, and I will stick on this. And thank you for your attention.